Okay, so welcome to this next video. In this video what we're going to talk about is high density lipoprotein and the reverse cholesterol transport pathway. Okay, and the reverse cholesterol transport pathway is the main pathway which high density lipoproteins are involved in. Okay, right. So I'm just going to give you the one-line sentence which tells you what reverse cholesterol transport is, basically. And then we're going to firstly discuss some prerequisites. Okay, so we're going to discuss, for instance, the structure of high-density lipoproteins, and we're going to make sure everyone is familiar with the different types of lipids contained within high-density lipoproteins. Okay, and then we'll move on to a in-detailed study of the pathway for reverse cholesterol transport. Okay, so the one-liner then that sums up what reverse cholesterol transport is then, and what the role of high-density lipoproteins are within the human body, is that it is the transportation of cholesterol from peripheral tissues to the liver. Okay, so if you have cells that have far too much cholesterol in, okay, so let's say we've got a cell here that has got far too much cholesterol, so this cell has too much cholesterol in it. Now cells all need a little bit of cholesterol because cholesterol is an essential component of the cell membrane, but if you have too much, that's not good either. So what happens is, if this cell has too much cholesterol in, and I'm wondering how I can denote that it's got too much cholesterol in, I might draw it blue just to somehow denote that it's got too much cholesterol. Okay, if you've got too much cholesterol in you, what you activate is all sorts of pathways, one that we're specifically going to look at, and this is going to activate the removal of the cholesterol from you, and you're going to put it into high density lipoprotein particles, which will then deliver it back to the liver. Okay, and the main target is the liver. However, HDL will also take it to steroidogenic cells, such as the cells of the adrenal glands, the cells of the testes, and the cells of the ovaries, if you're female. Okay, so basically that's what reverse cholesterol transport is. It's the removal of cholesterol from peripheral tissue and the transportation of it back to the liver. Okay, whereas normally what happens is that cholesterol is being transported from the liver to other peripheral tissues. Okay, so reverse cholesterol transport is the opposite process, where you're taking it from the peripheral tissues where there is too much present back to the liver. Okay, now, the place where reverse cholesterol transport is very heavily studied is in atherosclerosis, because in atherosclerosis, in the intima of your arteries, what you get is macrophages infiltrating into the intima of your arteries, and these end up taking far too much cholesterol into their structures, basically. Okay, so they become cholesterol overloaded, and this eventually kills them, and they start releasing the cholesterol into huge droplets of cholesterol that form within the tunica intima, and these are called cholesterol crystals, and this gradually produces this sort of growth on the side of the artery wall, basically known as an atherosclerotic plaque. So, HDL is very protective against atherosclerosis because it can remove the cholesterol from these cholesterol overloaded macrophages and try and prevent the propagation of the atherosclerotic plaque. However, we're going to study reverse cholesterol transport in its more general form, which is just a way of removing cholesterol from cholesterol overloaded cells. Okay, right, so we'll come back to the in-detailed mechanism of reverse cholesterol transport later. What we're going to start off with is some prerequisites. Okay, so we're going to start off by discussing the structure of a high-density lipoprotein particle. Now, before I go any further with discussing the prerequisites, what I would like to do is get over and done with my reference for this entire video. Okay, so the initial stuff that we're going to discuss, for instance, about lipid structures, okay, that's not from this reference. However, some of the later stuff about reverse cholesterol transport, you'll find it more difficult to find this in the textbooks, okay, and therefore I should include a reference of where I got the information from. So my reference is a review, and the lead author is Lucia. Okay, there are also some other authors on this review as well, but this person is the lead author. Okay, and this was written in 2014, so it's nice and up to date. Okay, right, so that's my reference for this. Right, so let's now go on to the prerequisites for a discussion of high-density lipoprotein involvement in the reverse cholesterol transport pathway, which is that we need to understand what a high-density lipoprotein actually is. Okay, so let's turn our attention to what a high-density lipoprotein actually is. So high-density lipoproteins are usually abbreviated to HDL for short, H for high, D for density, and L for lipoprotein. Okay, and the basic idea is that they are spheres of lipids. Okay, so let me draw one of these. So here is the sphere. Okay, and the outer surface of this sphere is going to be made up of phospholipids. Okay, so what you're going to have lining the outer surface of the high-density lipoprotein particle is a phospholipid monolayer. Okay, and by the way, this does go across, it does extend to other lipoprotein structures. Okay, so we're going to later see other examples of lipoproteins. Specifically, the other one we're going to see is low-density lipoprotein, but we'll come to that when we get there. Okay, but low-density lipoprotein has almost an identical structure to HDL. So for now, I'll just introduce the structure of HDL, and then we'll say how LDL differs from HDL later. Okay, right. So this outer shell of the lipoprotein structure is called the phospholipid monolayer. Okay, and that's because it's made up of a monolayer of phospholipids, so a single layer of phospholipids. So let me tell you about what the criterion that you need to satisfy is to be a phospholipid. In fact, it's not a criterion, it's criteria. So there are two criteria that you need to satisfy in order to be considered a phospholipid. Okay, so to be considered a phospholipid, within your structure you need to have a phosphate group. Okay, and you also need to have a long-chain carboxylic acid. So let me discuss the structure of these two things. Now, long-chain carboxylic acids are also often called fatty acids. So let me discuss what these two things mean. So let's start with a phosphate group. So basically, a phosphate group consists of a phosphorus atom at the center, okay, and then double bound to the phosphorus atom, what you have is an oxygen atom, and then also off the phosphorus atom you have two alcohol groups, one and then your second one over here. Okay, and then finally coming off the phosphorus atom and completing the phosphate group, you have a single bond to an oxygen atom. Now this oxygen atom has then acquired another electron via ionic means to take up its total outer shell electron number to 8, and that means that it has a negative charge there. Okay, so this is the structure of a phosphate group. Now this at the moment is well, actually it's not a phosphate group, this is the structure of a phosphate. Okay, this is the pure phosphate. When you bind it onto something, it will then be called a phosphate group. Okay, so you need to have one of these within your structure, and you also have to have a fatty acid. Now, fatty acids is the sort of colloquial name for what should really be called a long-chain carboxylic acid. Okay? And although long-chain carboxylic acid is a bit more of a mouthful than fatty acid, it's a useful name because it tells us exactly what we're dealing with, whereas fatty acid doesn't really. It tells us that we're dealing with a carboxylic acid molecule where we have an extremely long tail, basically. Okay, so here is the carboxylic acid group here. Okay, and then you're going to need to have an extremely long tail here, so I'll put R for an arbitrary group. So let me give you an example of a long-chain carboxylic acid. So the sort of prototypical example of a long-chain carboxylic acid is stearic acid. Okay, and stearic acid is the old biochemist's name for a molecule that now is more correctly called octadecanoic acid, although people still use stearic acid as a common name. Okay, and again, octadecanoic acid is a better name because it tells us once again exactly what we're dealing with. It tells us that we are dealing with a carboxylic acid which is fully saturated and has 18 carbons. Okay, so here's the carboxylic acid group. That's our first carbon. We then need another 17 carbons coming off this. Now 16 of these are going to be in the form of methylene groups. Okay, so I'll show these here. So here's a methylene group, and then we want to repeat that 16 times. So I'll put brackets around it like so, and then I'll subscript it 16 like so. Okay, and that means repeat this methylene group 16 times. That's a useful trick for getting out of having to draw 16 methylene groups. Okay, and then on the end of the final methylene group, what you then have is a methyl group coming off. So this now is the structure of stearic acid, or octadecanoic acid. It's an 18 carbon, fully saturated carboxylic acid. And it would be an example of a long chain carboxylic acid because we have a really long tail here. Now, that's all the criteria that you have to have to be called a phospholipid, basically. You need to have some sort of long-chain carboxylic acid within your structure and a phosphate group within your structure. Okay, now I'm going to discuss the different types of phospholipids now, but before I just do that, what I'd like to discuss is more generally what actually is meant by a lipid molecule. Okay, so what do we actually mean when we say a lipid molecule? Well, basically, lipid molecules are the more medical name for fat molecules. And the characteristic feature that fat molecules share is that they all have extremely hydrophobic structures. Okay, so they don't interact well at all with water. Now, why do they not interact at all well with water? Well, basically, they are usually neutral. Okay, now, water is an extremely polar structure, so let me show you the structure of water. So here is water. Okay, now in each of these covalent bonds between the oxygen and the hydrogen atom, you have 2 electrons. 1 electron from the oxygen and 1 from the hydrogen. Okay, so let's show these here. So we'll say that the dots are from the hydrogens and the crosses are from the oxygen. And the oxygen will also have 2 lone pairs of electrons in its outer shell, like so. Okay, right. Now, basically the electrons in these 2 covalent bonds here, do they sit right in the middle of the 2 atoms, i.e. the oxygen and the hydrogen atom, or do they sit towards one of the atoms, i.e. do they prefer being near one atom as opposed to the other atom? Okay, well basically the answer is that they prefer sitting near the oxygen atom. So basically both of these atoms here, the oxygen atom and the hydrogen atom, have nuclei, and those nuclei contain protons. In the case of the hydrogen nucleus, it only contains 1 proton. In the case of the oxygen nucleus, you have multiple protons. You then have, in the case of the oxygen atom, inner shells of electrons which shield the outer shell from the nucleus. Okay, but overall you then have to weigh up these electrons are feeling an attraction to the nucleus of this oxygen atom and also to the nucleus of this hydrogen atom. Okay, and then the question is, which one is going to pull on them harder? Well, there is a fancy word that means how hard a nucleus will pull on electrons which are within covalent bonds like this, and it is electronegativity. Okay, So we would say that oxygen has a greater electronegativity than hydrogen, and what that means is that the oxygen nucleus, even with these inner shell electrons which are shielding it from these electrons in this covalent bond, is going to exert a greater pull on the electrons within this covalent bond than will the hydrogen nucleus. Okay, so that's what is meant by electronegativity. The ability of the oxygen atom to pull electrons within a covalent bond is greater than the ability of the hydrogen nucleus to pull electrons within a covalent bond. Therefore, the electrons in these covalent bonds will sit more towards the oxygen atom than they will towards the hydrogen atom. Okay, and when you have both of these pairs of electrons in these two bonds sitting towards the oxygen atom, that will give the oxygen atom a partial negative charge, whilst it will give the protons a partial, the hydrogen atoms a partial positive charge over here. Okay, right, so that's what is meant by water having an extremely polar structure. You have an unequal distribution of charge, basically. You have a certain area which is more negative, and then areas which are more positive. Now, contrast that to an example of a lipid molecule that we have here, which is this long chain carboxylic acid here, stearic acid. Okay, stearic acid has this incredibly long tail of 17 carbons which are completely saturated. Now, that tail is extremely hydrophobic. Why is it extremely hydrophobic? Well, it's because the structure is very neutral. You don't have much unequal distribution of charge. Okay? What I need to tell you about is the fact that water will not interact well with this, because water interacts so well with other water molecules. So let me just explain something about the way that water molecules can interact with one another. If we take another water molecule here, okay, like so, then we know that this oxygen here has two lone pairs of electrons on it, and it has this partial negative charge because the electrons in these covalent bonds here are sitting towards the oxygen atom over the hydrogen atoms. Okay, and the hydrogen atoms have the partial positive charge. Okay, basically the partial positively charged hydrogen atom here can form a very strong interaction with this lone pair of electrons on the oxygen atom over here. Okay, so let me show this. So this very strong interaction is known as a hydrogen bond. Okay, and it is a form of intermolecular bonding. However, it's a very strong form of intermolecular bonding. So water molecules have these very strong interactions between one another. Now, if we look at this stearic acid molecule is very neutral. Now, the reason it's very neutral is that it only consists of bonds between carbon atoms and hydrogen atoms. Carbon definitely has the same electronegativity as another carbon atom, so you're not going to have unequal distribution of the charge within these covalent bonds between carbon atoms. In addition, hydrogen has roughly the same electronegativity as carbon, so the electrons will be sort of sat in between the two here as well. So you will just have a neutral molecule, apart from this head, but the main bulk of the molecule is just extremely neutral. Now, this is the reason lipid molecules don't interact well with water. They have these incredibly neutral structures. Now, if we bring a water molecule and ram it against one of these extremely hydrophobic tails here, I mean, it will interact with it, but it won't interact with it very strongly, okay? So, if the water has a choice, which of the two will it interact with? Another water molecule or this extremely neutral molecule? Well, it will always pick the water molecule because it can form a much stronger interaction. So when you have a huge amount of water and all the water molecules are interacting with one another, it's very difficult to dissolve fat molecules in that water because you would have to break the bonds between the water molecules, okay, and then you would produce new bonds between the water molecule and the fat molecule, and basically that's just not thermodynamically favourable, because breaking the hydrogen bonds between water molecules is going to require a lot of energy, okay, and the amount of energy you're going to then release when you bind the water molecules onto the long chain carboxylic acid is going to be very small. So overall, you're going to go from a low energy state to a high energy state, and that's just not thermodynamically favourable. Okay, and that's why water and fat don't really mix at all. If you do try and mix fat with water, what happens is the two of them separate into two distinct layers, so that you can absolutely minimize the interaction of water with the fat molecules. Okay, so basically all of the fat molecules get pushed out of the way so that the water molecules can continue to interact with each other. So it's not that the water molecules don't like the fat molecules, or that the fat molecules don't like the water molecules. It's that the water molecules would just rather be with other water molecules than with the fat molecules, so they just sort of push them out of the way, basically. Okay, right. So that's just a little bit of an insight into what lipid molecules actually are. They're these hydrophobic molecules that have equal distribution of charge and are therefore neutral and don't interact therefore at all well with these very polar water molecules. Okay, right. So now let's go on and discuss examples of phospholipids, okay, because there are two major classes of phospholipids that are important within biology. Okay, so the first class is the class that you most likely think of when you think of a phospholipid. Okay, so people generally have a picture in mind when they think of a phospholipid, and generally they will be thinking of a phosphoglycerolipid rather than the other type of phospholipid which we'll come to. Okay, so we'll start off by discussing the structure of a phosphoglycerolipid. Now, as the name suggests, these phosphoglycerolipids are all going to have glycerol within their structure. Okay, now, all of the phosphoglycerolipids are going to be based on the structure of a molecule called phosphatidic acid. Okay, so I'm going to start off with a discussion of what phosphatidic acid is and the structure of phosphatidic acid, and then we'll come on to how you can modify phosphatidic acid and produce a more general phosphoglycerolipid. Phosphatidic acid itself is a phospholipid, and it is considered a phosphoglycerolipid, but it's kind of the core phosphoglycerolipid, and all the others are then derived from it. Okay, so let's have a look at the structure of a phosphatidic acid molecule. Okay, so we're not going to draw the full formula, we're going to draw a little cartoon. So let me draw a cartoon for this. So, these horizontal lines here will represent two long-chain carboxylic acids. We're then going to esterify them to this vertical line here, which is going to represent a glycerol molecule. And then off the third carbon's alcohol group of the glycerol molecule, you're going to have a phosphate group. And that's the structure of a phosphatidic acid molecule. So let's now discuss this in a bit more detail. So, we'll start by colouring it in. So, in orange here, these are two long-chain carboxylic acids, and for short I'll abbreviate long-chain carboxylic acid to Fa for fatty acid. Okay, so these are long-chain carboxylic acids. Now, this is the hydrophobic tail here, and then right at the tip here, you'll have the carboxylic acid group of both of them. Now, I should stress that they don't have to be a set long-chain carboxylic acid. They can be any old long-chain carboxylic acids, and in addition, they don't have to be the same one. So the one that you have here doesn't have to be the same as the one here. I know I've drawn them the same length, but they don't have to be. They can be different long-chain carboxylic acids. Okay, and they've had their carboxylic acid groups esterified onto the alcohol groups of the first and second carbons of the glycerol molecule here, which is being represented as this vertical green line here. Now, again, glycerol is the old biochemist's name for a molecule that has since acquired a new name, and its new name is propane 1,2,3 1,2,3 trial. And although propane 1,2,3 trial is a bit more of a mouthful than is glycerol, it's a useful name because it tells us exactly what we're dealing with here. It tells us that we're dealing with a 3-carbon molecule where you have alcohol groups coming off the first, the second, and the third carbons of that 3-carbon molecule. Okay, so that's propane 1,2,3 trial. Now, let's just discuss, just to remind you, how you esterify long-chain carboxylic acids onto the alcohol groups of a glycerol molecule. Okay, so let's take our long-chain carboxylic acid here. So here's our carboxylic acid group with the R group here, so that's some long tail. Okay, and then we've got the alcohol group of the glycerol molecule here, and we want to attach this long chain carboxylic acid onto that alcohol group of the glycerol molecule via an ester link. So basically the way that you do this is you remove the alcohol group from the carbon of the carboxylic acid group, you remove the hydrogen from the alcohol group on the glycerol molecule, you then bind the carbon of the carboxylic acid molecule to the oxygen of the alcohol group, okay? And that then is called an ester link. Meanwhile, you attach the alcohol group and the hydrogen together to produce water, which is why esterification is considered a condensation reaction, because it produces water. Okay, so this is how you esterify long-chain carboxylic acids onto the alcohol groups of the first and second carbons of the glycerol molecule. Okay, so that gives us what's known as a diacyl glycerol. Okay, so when we've got 2 long-chain carboxylic acids attached to a glycerol molecule, that's just called a diacyl glycerol. We now want to attach a further structure onto the alcohol group of this 3rd carbon down here to turn it into what's known as phosphatidic acid. Okay, and in purple here, you might be able to guess what this is, because I told you that phosphatidic acid was an example of a phospholipid. Okay, so we've got a fatty acid in our structure. What was the second criterion that you needed to obey in order to be considered a phospholipid? You needed a phosphate group. Okay, so this is going to be then a phosphate group attached onto the alcohol group of the 3rd carbon of the glycerol molecule. Okay, so let's just discuss how phosphate groups can be attached onto alcohol groups. Okay, so let's draw out the phosphate group structure again. So here is the phosphorus atom double bound to the oxygen. Then we have these 2 alcohol groups coming off the phosphorus atom, and then we have a single bond to an oxygen atom, which has then acquired another electron via ionic means, and therefore has a negative charge here. Okay, so let me get my highlighters out, and let me highlight this structure here. Okay, what does that look like? We have a phosphorus atom here, double bound to an oxygen with an alcohol group coming off. Well, it looks hopefully very much so like a carboxylic acid group. In fact, if I change that phosphorus atom to a carbon atom, ignore the fact that it's got 5 bonds coming off, which is absurd for carbon, and just look at the blue structure. If I change that to a carbon, we do indeed have a carboxylic acid group there. Okay. So, in actual fact, this structure can interact with alcohol groups in pretty much the same way as carboxylic acids can interact with alcohol groups. So if I bring an alcohol group in here, what can happen is the alcohol group can come off the phosphate group, the hydrogen can come off the alcohol group, those two can combine together to make water. Okay, so these will combine together to make water, and then the oxygen atom of the alcohol group combined with the phosphorus atom of the phosphate group to make something that's very similar to an ester link. Now it's not the same, so it's called instead a phosphoester link. Okay, right. So that's how we're going to attach a phosphate group onto the alcohol group that comes off the 3rd carbon of the glycerol molecule. Okay, so that then now is the structure of phosphatidic acid. Now, before we go any further and discuss how you go from phosphatidic acid, which is the core phosphoglycerolipid, to other phosphoglycerolipids, what I want to firstly discuss is the difference between phosphatidic acid and another molecule known as phosphatidate. Okay, so I want to discuss what is the difference between phosphatidic acid and phosphatidate, because you'll more commonly hear people referring to phosphatidate rather than phosphatidic acid. Okay, so basically, phosphatidic acid is an acid because it can donate protons into solution. That's the criterion that you have to obey in order to be considered an acid. You have to donate protons into solution. Okay? Basically, this alcohol group has gone now. This is involved in the phosphoester link to the alcohol group of the third carbon of the glycerol molecule. However, this alcohol group here can actually lose its proton, basically, and then this oxygen will be left with a negative charge. Okay, so when the proton cleaves off, it will go into solution, and therefore the phosphatidic acid molecule has acted as an acid by donating a proton away into solution. Okay, so that's why phosphatidic acid is considered an acid molecule, basically. The structure that you get once the phosphatidic acid molecule has donated its proton away. So imagine this proton has fallen off now, and this oxygen has a negative charge here. That structure that you end up with is known as phosphatidate. Okay? It is no longer an acid, because it has no proton which it can donate into solution. An acid is a molecule that is capable of donating a proton into solution. Once you've donated the proton away, you can't donate it away again, basically. Okay, so phosphatidate is the conjugate base of phosphatidic acid. And this is a general principle for all acids, that once they donate their proton away, they end up with a negative charge. Okay, they are no longer an acid because they can't donate another proton away, in most cases at least. Okay, and instead they've actually become a base, because they, instead of being able to donate a proton away, instead they're actually able to receive a proton out of solution. The proton can come and bind to the group that has the negative charge, basically. Okay, so they go from being an acid to being a base when they lose their proton, basically, and they are then called the conjugate base of the acid. Okay, right, so that's the difference between phosphatidic acid and phosphatidate. Now, under physiological pH, which is around 7.45, the phosphatidic acid molecules generally do donate their protons away into solution, and therefore most of them generally exist then as phosphatidate molecules rather than as phosphatidic acid molecules, which is why people generally talk more about phosphatidate than they do about phosphatidic acid. Okay, because most of the molecules will be present in the conjugate base form at physiological pH. Okay, right, so that's the structure then of this phosphatidic acid molecule. How do we go from that to a more generalized phosphoglycerolipid? Well, basically, let me get my highlighter again. Okay, we've had this alcohol group here cleave off and join with that hydrogen, and you formed this first phosphoester link. However, even once you've done that, you've still got this group here intact. You've still got a phosphorus atom double bound to an oxygen with an alcohol group coming off it. Okay, which is exactly the same as the group that we originally had circled in blue. Okay, so this is actually capable of interacting now with another alcohol group, which is something that carboxylic acid groups can't do. Phosphate groups effectively are better at doing what carboxylic acids do than they are themselves, because they have two of these structures basically which can bind onto alcohol groups. So what you can do is you can take another structure with an alcohol group on it, and you can link these two together. You can link this phosphate group that's already attached onto the phosphatidic acid molecule. You can link it onto this other molecule which has this alcohol group by taking the alcohol group off the phosphate group, adding it onto the hydrogen off the alcohol group, and then binding this phosphorus atom to this oxygen atom here to create a phosphoester link. So we're going to stick a group on there effectively, and this is going to have to be an alcohol molecule that we're sticking on there, and that's how you get to the more general phosphoglycerolipid. You stick an additional group onto that spare alcohol group that comes off the phosphate group of the phosphatidic acid molecule. Okay, right, so I want to show you now an example of a molecule that you can attach onto this phosphate group of the phosphatidic acid molecule. Okay, and the example that I'm going to give you is choline. Okay, so let's go on to the other side. So, basically what we're going to do is attach on an alcohol called choline, and the molecule you overall get then is going to be called Phosphatidylcholine. Okay, and this is a general principle for naming phosphoglycerolipids. They all start with the prefix phosphatidyl. Okay, and what that shows you is that within the structure you have a phosphatidic acid molecule, basically. Okay, so that's referring to all of this. It's referring to the long-chain carboxylic acids, the glycerol molecule, and then the phosphate head. And by the way, it might be a good idea now to stress that there is not just one phosphatidic acid molecule. There are many different phosphatidic acid molecules, because there is not just two long-chain carboxylic acids that you can stick in these positions. These are not set in stone, basically, and you can vary them, and therefore phosphatidic acid is actually a whole class of molecules that have very similar properties, because exactly which long-chain carboxylic acids you have here aren't really going to influence what these molecules can actually do. Okay, so that phosphatidyl prefix then refers to the phosphatidic acid molecule that's within the structure, and then the choline refers to the alcohol group that has been added onto the phosphate group of the phosphatidic acid molecule via a phosphoester link. Okay, now choline is the same molecule as you have within the famous neurotransmitter acetylcholine. In acetylcholine, what you do is you esterify acetic acid, which is the old name for ethanoic acid, a 2-carbon fully saturated carboxylic acid. You esterify that onto choline, and that gives you acetylcholine. Okay, in this case we are going to phosphoesterify choline onto the phosphatidic acid molecule. So let me now show you the structure of the choline alcohol. Okay, right, so you have an alcohol group, clearly. Then what you have is an ethylene group, which basically consists of 2 methylene groups bound together, like so. And then off the other side of the ethylene group, what you then have is a nitrogen atom, which will have 3 methyl groups coming off. So 1, 2, and then 3. Now that leaves this nitrogen of the choline molecule with 4 bonds coming off it. Now, nitrogen does not like having 4 bonds. It's meant to have 3 bonds and then a lone pair of electrons. So the fact that it has 4 bonds means that in one of these bonds, what you have done is you have put both of the electrons that are going to make up the covalent bond from the nitrogen. Okay, so let's say it's this bond here that's got this sorted arrangement going on, and basically what has happened is the nitrogen has put in both electrons, basically, and the carbon hasn't put in a single thing. Okay, so the usual understanding of a covalent bond is that 1 electron comes from each member, okay, 1 from the nitrogen and 1 from the carbon. So when the nitrogen puts in both, it's as though the nitrogen has given away 1 to the carbon, because the arrangement is that this one belongs to the carbon and this one belongs to the nitrogen, basically. So when the nitrogen does this, it's as though it's given it away to the carbon, and then the carbon has sort of played this horrible trick on it and said, right, now let's form a covalent bond. Yes, I had this electron all along, let's do it. Okay, and they then form a covalent bond. So that means that the nitrogen gets a positive charge because it has lost an electron. And you might say, well then does the carbon have a negative charge because it's gained an electron? And the answer is no. And the reason is that when the carbon came into this horrible deal here, it would have originally had a positive charge, so it would have gone and irresponsibly lost an electron at some point, and it's then come along and nicked an electron off the nitrogen. It's neutralized itself, but it's deferred the positive charge onto the nitrogen, and that means that the whole choline molecule does in actual fact have a positive charge. Okay, so when you link this molecule here, which is choline, onto phosphatidic acid, you get phosphatidylcholine, which is often abbreviated to PC, and this is also called lecithin. Now, phosphatidylcholine is an example of a phosphoglycerolipid. It is not the only phosphoglycerolipid. There are loads of other alcohol groups that you can add on to that phosphate group of the phosphatidic acid molecule. However, lecithin is an extremely important example of a phosphoglycerolipid, as we'll see later. Okay, so when you esterify phosphosterify choline onto the phosphate group of a phosphatidic acid molecule, the phosphoglycerolipid that you then get is phosphatidylcholine, and this is an extremely important example of a phosphoglycerolipid, and it's got this other name, lecithin. Okay, right, so we'll call it there for this video, and in the next video what we'll do is look at the other examples of phospholipids that are important within biology, which are the sphingomyelins.